Good afternoon. I would like first to present myself. I'm a biomedical engineer. I have 7 years research experience in tissue engineering, designing materials, understanding and controlling the interactions of the cells with that structure so that the cells do their function, the function we want them to do. And I've been working with additive manufacturing for 10 years. 4 of them apply to food technology at INL. And my talk will be about some of our approaches to boost foods using additive manufacturing. I will briefly present INL as well. INL is the only intergovernmental institute for nanotechnology with the state-of-the-art infrastructures that counts with the representation in many countries. And we try to integrate the worldwide best solutions to the commercial needs, creating value to the society. We are organized in 6 clusters where 24 research groups are integrated to provide the best solutions in that particular areas. And among many things, we develop sensors, batteries, and food solutions. In the food processing groups, we are organized in 3 research lines, functional foods, food packaging, and food personalization, focused to solve the most challenging aspects of the food industry. So in the functional foods, we replace unhealthy fats in meat products using oleogels. We research society regulator compounds, and we also incorporate antioxidants and a wide variety of bioactives, edible bioactives using several technologies. We developed also biodegradable, active, and edible food packaging, and for instance, prototypes like super hydrophobic thermoinsulated paper bags for fresh foods. And in this scope, we also have a recent spin-out called To Be Nanofoods. In the view of personalizing the future foods, to study digestibility and other aspects, we created a gastrointestinal tract on a sheep. We explored 3D food printing and also its application to develop the meat alternatives. And this is a slide that I've made some years ago, and it has not radically changed yet. It seems that consumers are not ready to have food printers at home. At least I don't know anyone. We still look for applications and still need new and tailored printers. And why is that? So in my opinion, technology is not ready to fit in the routines of consumers. Is not yet a tool answering to their demands or ready to prepare the dishes of the whole family in a few minutes, let's say. And to get there, we need to tailor the systems for the ideas according to the ingredients needs or limitations, the 3D models, and the final properties that the product has to have, and also improving the production time. And that should be done for both cases of consumer end or industrial application, and that means we have a lot to be done yet in both consumer, nutrition, and the technology sides. I will now share with you some works we have been doing to advance additive manufacturing of foods. Our research is focused on solving the biggest challenges of food industry, so food personalization at the level of composition, texture, nutrition, and shape. So, and for that, we need to learn about the nature and composition of the inks, how to formulate, how the ink's properties influence the printing, and to understand how to control the final properties of the products. And here in this study, We developed two-phase inks, so by phase separation of whey protein isolates, which is a functional protein, where we can alter the structure of the proteins from fibrillar-like to particles, and also the the biological properties. They are highly shear thinning, they are extrudable at low pressures, which is important to avoid reaching the printer, I call it printer pressure dead zone, if we want to increase the speed, which is important for the scalability of this technology. And among other properties, we have estimated shear stress of, to the wall for different printing conditions and correlated the structure and biological properties with the printability. One of the challenges with food's functionality that you might be aware of is in preserving the bioactive properties during processing and, for instance, during baking. So our hypothesis here was that by controlling the printing variables and baking, we could improve the bioactivity, and in this case, we used cookies enriched with encapsulated polyphenols obtained from grape skins. And we observed a 20% improvement of the antioxidant activity by encapsulation. And the novelty that was observing an increase of 150% for a particular for a particular combination of a number of layers, infill, baking time, and temperature. We saw that independently of the porosity, the antioxidant activity dropped with the baking time. So what happened was that by introducing pores and reducing a little the thickness, the moisture dropped faster, triggered the Maillard reaction earlier, which avoided an extensive loss of the antioxidant activity. And one of our tasks is also to find the best solution to reverse engineer the bovine or other meats, producing cell-made meats with proteomic, nutritional appearance, and organoleptic properties similar to the slaughtered meat. The solution will require customization, cost effectiveness, a high production speed, scalability, the use of only edible and food grade ingredients and processes, and also a high regulation of cell behavior. So we could be able to, at an industrial level, produce all kinds of trimmed and bone meat cuts like a single animal can do. And we see 3D food bioprinting as the best candidate to accurately and reliably reproduce the whole complexity of meat, and that can fulfill all the requirements mentioned. So it starts from a computer-assisted design, so we can program what is deposit, what is the composition, and where the printed units that can be spheres or filaments are deposited in each of the layers that is built up. The technique allows multi-scale control of the final products, and a wide variety of edible inks, cells, and process parameters give us the chance of making almost an infinite number of products from only one machine. You may wonder why we call it 3D food bioprinting. So we are working with foods and living cells, and the final goal is to produce a safe meat product instead of a functional and highly complex tissue, as is done with bioprinting and tissue engineering. And 3D printing can be used in 3 different ways to produce meat alternatives. So a cellular, using 3D food printing, like to do a plant-based meat, or 3D food printing combined with cellular integration in a way not directed by the 3D printer, which corresponds to 3D printing of biomaterials in tissue engineering. And the main difference between that with the 3D food bioprinting is that with this one, we control the spatial deposition of the cells that are landed inside the inks with the printer. And the flow of the process does not depend on cell attachment to any surface and can be done at very high cell densities. However, it comes with the challenge of overcoming printing speed and consequent process efficiency and scalability that is more challenging here. And from the perspective of a centralized cultivated meat process, like a continuous one, seeking high efficiency, the printer is can be seen in the printer is the center point, and the printing resolution and the maximum speed come as the primary process constraints that will determine the production flow. Those parameters will translate as a specific cell harvesting rate and inks production that have to match the flow of the edible ink required to feed the printer. So it seems straightforward, but high speed and high resolution comes with a no. So there will be a limit which cell viability and maturation versus productivity will not be balanced anymore. And with MITIT, that is a project that was granted us by the Good Food Institute. We work on the root of some of those challenges. We explore how this technology can be applied for cultivated meats and developed a bioprinted model to study and control the appearance, the texture, cell behavior, and trying to maximize the production flow. It consists in formulating plant-based inks, cell studies, food bioprinting, and considering multiple variables that might affect the texture. And one of the variables important is resolution. Is important for the production volume, and it determines the maximum speed at which we'll be able to print. It will undoubtedly influence the appearance, and it might also change the eating experience and consequently final consumer acceptance. And you can see the difference between one sample produced with high and the other with low resolution. And here there are many decisions along the process. I believe that this will be the most difficult technology to use here, and many decisions that the project is not considering have to be taken, and we have a lot of decisions to be balanced and process parameters to be optimized. And I close by inviting you to submit research or review manuscripts to the special issue we are co-editing, bio-based additive manufacturing in food and tissue engineering in polymers. And also by acknowledging everyone that has been involved in the research and the Good Food Institute for the funding. Thank you for your attention.